Dames en heren, het cortège.
Mr. Rector Magnificus, Your Excellency, the Ambassador of Malaysia, Datu Dr. Fauzia Mohammed, esteemed members of the audience. Those of you well versed in Darwin have of course realized that the words endless forms in the title of this inaugural lecture are a quote from the last sentence of On the Origin of Species, which reads, "There is grandeur in this view of life, with its several powers, having been originally breathed into a few forms or into one. And that, whilst this planet has gone cycling on, according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." I am not the first to have mined this sentence for quotations. Far from it. Stephen Jay Gould's long-running column in Natural History magazine was named This View of Life. Sean Carroll's wonderful book on Evo Devo was titled Endless Forms Most Beautiful. And I too, with this lecture, have yielded to the temptation of quoting from this poetic coda to the greatest work in biology, in the title, but also throughout this inaugural lecture. This professorship is hosted within the Faculty of Mathematics and Natural Sciences. But biology has always shifted rather uncomfortably in the pews among the other sciences. For its chief source of inspiration, the diversity of life, is also its greatest handicap. Mathematics, chemistry, and physics are characterized by the quest for universalities. And while many biologists will profess that the same is true in biology, this aim is constantly thwarted by the fact that many biologists' inspiration and motivation lie more in life's diversity than in its unity. Naturalis Biodiversity Center, for instance, is a temple in veneration of this disparity and diversity in the manifestations of the living world. The endlessness of the forms in which life displays itself is so overwhelming that many biologists take leave of any desire to contribute to the discovery of universal laws and instead identify themselves by the narrow group of organisms that they study. Microbiologists, ornithologists, and botanists, but even coleopterologists, copepodologists, and coptoclaviskinologists form their own separate departments and professional societies. They have their own conferences and publish their own journals. While this fragmentation is necessary and understandable, it has hampered the search for universalities in biology. As Stephen Hubble, an ecologist who has forged a universal theory of ecology, has lamented, if Galileo had been a biologist, he would have spent his whole life throwing all kinds of objects off the Leaning Tower of Pisa and never have come up with the concept of gravitational acceleration. And yet, despite this handicap for the furthering of biological unification, biologists' infatuation with diversity is the inalienable foundation of the field. Yes, genomics, cell biology, and developmental genetics of model organisms like the zebrafish Danio, the tailcress Arabidopsis thaliana, or the fruit fly Sophophora melanogaster will yield deep insights for those particular species, but these insights will gain much in importance by confronting them with the touchstone that the full biodiversity offers. This is why this chair for character evolution and biodiversity is so crucial, because it connects the Institute of Biology Leiden, IBL, which, via the groups of, for example, Marijn van der Zee, Mike Richardson, Herman Spink, and Peter Klinkhamer, has seen an increasing focus on model species, with, on the other hand, Naturalis Biodiversity Center, where the focus has always been on entire communities, entire clades, and entire ecosystems. I say connect, but I should really say reaffirm the connection, because Naturalis and the university have had a long and checkered history of approach, and sometimes also repulsion, beautifully described by L.B. Holthuis in his book about the history of the museum. One of the persons in whom the IBL Naturalis connection was most recently embodied is my former MSc supervisor, PhD advisor, and professor of systematic zoology, Edmund Gittenberger. In many ways, I pride myself on continuing the research on character evolution in snails that he began with his projects on Albinaria and other snail taxa. And I'm eternally grateful to him for developing a low-tech but high-impact data visualization device called the opplakkaart. Representative land snail shells are glued onto a map of, say, the Greek island of Crete, and in one glance, the full breadth of geographic differentiation in shell morphology becomes visible. Moreover, it is immediately apparent that dramatic evolutionary patterns exist at very small spatial scales. Sometimes shell features are seen to disappear along the few kilometers between one village and the next. One of my formative moments as an evolutionary biologist was to walk through these evolutionary patterns during fieldwork in the mountains of Crete and to literally see the snails change shape with every step I took. The tendency for land snails, due to their low mobility, to display such impressive evolutionary patterns on very small spatial scales makes them ideal subjects for studying diversification and endemism. This is exemplified by our work on microsnails on isolated limestone outcrops in Malaysia and Borneo, featured only last week in the successfully defended PhD thesis of my student Liu Torsung, who, tomorrow, will return to his post at University Malaysia Sabah. Dr. Liu's work, besides trailblazing new approaches to shell morphometrics and cyber taxonomy, has shown how the fragmented nature of the habitat results in microgeographic patterns of endemism. Numerous Southeast Asian microsnail species live exclusively on isolated limestone hills, many of which are disappearing rapidly because they are quarried for cement. This reveals an urgent conservation concern. Several species in peninsular Malaysia are already extinct, many seriously endangered, and one of the new species discovered and named by Dr. Liu, Plectostoma tengekensis, of Batu Tenggek in the state of Pahang, will even go extinct before the end of this year, the very same year in which its official description was published. Not only are snails eminently suited for visualizing microgeographic diversification, but also for elucidating the forces driving it, thanks to the fact that their shells, including any selection-induced damage, remain in the environment for long after the snail has died. Moreover, the shells of living snails are easily marked and retrieved individually, allowing manipulation and mark recapture studies. This helps us to study, for example, adaptation to predators, and the work of several of my students has enabled us to outline a research program for studying and quantifying the selection pressure imposed by predators. Not just Dr. Liu's work on Plectostoma and its beetle and slug enemies, but also the work by my students Renate Helwerda, Renze Schelvorst and Els Baalbergen. With support from the Jan Joost ter Pelkwijk Fund and the Uitenbogert Eliasen Foundation, they performed a natural history study on the drillus beetles that prey on Albinaria and Sepea snails. This work was carried out at field sites in Greece, in collaboration with Dr. Zinos Jokas of the University of Patras, and in IJmuiden, in the Netherlands. And their work was published yesterday in the journal PLOS ONE and has the dubious honor of being featured on Fox News this morning. Again, applications in conservation exist. After mentioning our research on my Twitter feed, we were approached by the municipality of Velzen to help adapt the restructuring of the old railroad along the Noordzeekanaal, one of Els Baalbergen's field sites, and Velzen has been consulting her to safeguard the survival of the local population of Drillus flavescens. Sepea shell color, a classical system for ecological genetics, is another character for which we will continue studying the evolution. Thanks to the citizen science project Evolution Megalab, which I will mention later during this lecture, the study of Sepea has seen a recent revival, with several research groups in Europe applying novel methods to this old system. In Naturalis, we have employed our collections to show small-scale thermal adaptation to climate change in the Netherlands, a study we published in the journal Global Change Biology, and incidentally a good example of how important it is for Naturalis to preserve large samples of common species and never ever to throw anything away. In another Sepea study, published in Heredity, we've made use of the known age of the snail populations in the reclaimed IJsselmeerpolders to measure the rate of evolutionary adaptation and determined that Sepea snails really are the Dutch Darwin's finches. Their colors evolving at the same rate as the beaks of these iconic birds, namely in the order of 20 kilodarwin, which is a measure for evolutionary change. And lastly, for the past few years, first-year biology students have done yearly monitorings of Sepea shell color in a population in het Groene Hart, which we maintain as a long-term character evolution monitoring project. The useful properties of snails as field experimental systems also allow us to employ them in one of my main research programs, namely the evolution of asymmetry. Although many organisms are fundamentally bilaterally symmetric, it is very common for them to evolve partial or entire asymmetry, either externally or, more frequently, internally. Once asymmetry has evolved, in theory, two mirror image forms are possible, and in reality, species may be monomorphic or dimorphic for these mirror image forms. Since this is a simple two-state character system, it is an ideal subject for finding general evo-devo patterns across unrelated taxa. Thereby, it cuts across the taxon-based subfields and may help find some of those elusive universal patterns in biology that I mentioned earlier. This is why, in the focus group Character Evolution, left-right asymmetry has been a major emphasis for the past years and will remain so for the years to come. Snails, like this Busicon carica from the Naturalis mollusk collection, courtesy of Jeroen Goud, are the ultimate asymmetric animals. Their body shape as well as their shell twisted into a helix. They can be dextral, like this one, which means they are clockwise coiled, with the aperture on the right-hand side if the tip is held upwards, or they can be sinistral, anti-clockwise coiled, like this species Busicon contrarius, with the aperture on the left when the tip is held up. Normally, like in these two whelks, species are monomorphic. A species is fixed for one mirror image form. Very rarely, however, we find snail species in which both mirror image forms exist side by side. The Malaysian snail Amphidromus inversus is one such exception that my students and collaborators and I have been studying intensively in the past 10 years. Lab and fieldwork have revealed that the dimorphism is maintained by the fact that, unlike the situation in most other snails, Amphidromus has a preference to mate with a partner that is its own mirror image. A curious situation that involves the mechanics of asymmetric genitalia and a corkscrew-shaped sperm package. In whole-body asymmetric animals such as snails, asymmetric reproductive organs are not unexpected. However, it is an unsolved mystery why asymmetric genitalia are so common in other animals as well. I carried out a study published in Animal Biology, which showed that the phenomenon is very widespread in chordates, including mammals, arthropods, nematodes, flatworms, and at least 6 other animal groups. The ubiquity of this phenomenon, as well as the conundrum posed by the fact that sexual characters are normally selected for symmetry rather than asymmetry, make this a very urgent and fascinating problem in character evolution. My students Rick van Beek, Tamara Hogeboom, Paulien de Jong, Melanie Meijer-Zoeslochtern, Tijmen Breeschoten, and Ruben Vijverberg have been addressing this problem in colevid and dynastid beetles, and my current MSc student Floor Rebergen has, with funding from the Kees Bakker Award, been able to pursue this in the group of Virginie Orgogozo in Paris, where a symmetric strain is maintained of the normally asymmetric Drosophila pachyia. Asymmetry in reproductive organs is not just restricted to animals. My colleague Barbara Gravendeel of Naturalis and the Leiden University of Applied Sciences has developed a branch of this research program on the evolution of asymmetric flowers in orchids, such as these, which have just been planted here by Rogier van Vught from the Botanical Garden. And these are 3 species of orchids, Prostechia cochleata, Dendrobium antenatum, and Paphiopedilum philippinense, which all have asymmetric flowers. One of these, this one, is a slipper orchid. Now, slipper orchids have twisted petals, and across the midline these can twist in opposite directions, clockwise on the left and anti-clockwise on the right, or vice versa, producing a symmetric flower, or they can twist in the same direction, either clockwise or anti-clockwise, resulting in asymmetric flowers. An evolutionary tree of Southeast Asian slipper orchids, reconstructed on the basis of DNA sequences by Malaysian PhD student Izan Ramli, revealed that petal asymmetry patterns show variable rates and directions of evolution, and that asymmetry and reversal to symmetry have evolved repeatedly. These orchids do not offer a reward to pollinators, and Gravendeel's team is now investigating whether the asymmetry may make the flowers harder to remember, so that it takes longer for pollinating insects to start avoiding them, long enough for them to be deceived at least twice, when they remove and again when they deposit pollen. So far, I have spoken about external asymmetries. But as our gut feelings can tell us, asymmetries are much more common in internal organs. One explanation for the evolution of asymmetry in internal organs is that it allows division of labor between left and right, such as is the case with the different roles of brain hemispheres in humans and other vertebrates, leading to lateralized behavior. And this brings me to the following audience participation exercise. Who of you would consider themselves left-handed? Okay, that's 13.4%, which is exactly the left-handed rate that is found in humans. Now next, would you please fold your arms, like so. Now who of you have their left wrist on top of their right arm? If so, please raise your hand. As you can see, in arm folding, the percentages are different. About 60% of people fold their arms consistently with the left wrist on top, and there is no correlation with writing and drawing-handedness of those same people. So if just now you answered that you are left-handed, this does not say anything about how you are folding your arms. Now finally, clasp your hands like so. Who among you have their left thumb on top? Thank you very much. Again, you fit with general surveys, namely 60% left thumb on top, 40% right thumb on top. But interestingly, in hand clasping, there is a correlation. If you are right-handed, you are slightly more likely to clasp your hands with the left thumb on top. You may relax now. This concludes the audience participation. But it does show that human-handed behavior, which is linked to brain laterality, is a much more complex trait than many people realize. And genome-wide association studies have recently shown that at least 40 different genes are involved. This complexity is further compounded by cultural evolution. In 2002, Karel ten Kate published a study of more than 1,000 Dutch and German university professors' portraits, including the ones on display in the Senaatskamer directly above our heads, in which he showed that the predominance of left cheek or right cheek preference in portraiture has changed over the centuries. This article appeared under the wonderful title, Posing as Professor, which is exactly what I and those in the benches are doing right now. Such links between morphological asymmetry and handed behavior provide an additional layer of relevance to a left-right asymmetry framework. And in this context, I could mention the work that César Rauch and Charles Franssen did on claw asymmetry in Pontonian shrimps, or Tom van Doren's work on mouth asymmetry and handed behavior in cichlid fishes. The evolutionary dynamics of asymmetry even has parallels in chirality dynamics in physics, like the work by Vincenzo Vitelli here at Lyon on crystal orientation in toroidal liquid crystals, or the work in the solid-state chemistry group of Elias Vlieg in Nijmegen. However, I believe I have amply showed the potential for this research thread, and for more detail, I would like to refer you to the asymmetry video made by Joris van Alphen for the focus group Character Evolution, which will be shown during the reception. Ladies and gentlemen, all too often it is said that an evolutionary view on life's diversity strips it from its magic, exposes its ugly bare bones, and removes all beauty and mystery. Indeed, evolutionary biology is expanding its reach into territories where previously other academic disciplines held sway. Evolutionary biologists dissect the ways in which our physiology has evolved to generate spiritual feelings, or love and compassion, or how moral judgment has been hardwired in our brains as an adaptation to living in tight-knit groups, meaning that human morality is not fixed, but has evolved and will continue to evolve. We tackle the evolution of everything that makes life worth living. And yet, I wholeheartedly agree with Darwin that there is grandeur in this evolutionary view of life. Personally, I much prefer a view of life in which I can marvel at the power wielded by eons of time, ever-changing selection pressures, and the accumulation of genetic change. To me, understanding brings the joy of belonging, and hence, is more gratifying than mystification. As Neil deGrasse Tyson emphasizes in his hit TV series Cosmos, a sense of being part of the fabric of life is much preferable over feeling like an awestruck outsider. It is this sense of investigative wonderment that I try to instill in the students that I teach in my lectures on systematics for first-year bachelor students, the orientation course Evolution, Biodiversity and Conservation for master students, and the international field course on tropical biodiversity for master students in Malaysia and Borneo. But not only in my students, also in the general public. 2 years ago, the Malaysian conservation organization Sabah Parks and Naturalis jointly organized the Kinabalu Crocodile Scientific Expedition. Some 20 Malaysian scientists and 20 Dutch scientists did DNA studies on endemic species on the summit of Mount Kinabalu, the tallest mountain of Southeast Asia. As revealed by the ongoing work of team members Casper Hendricks, Rimi Repin, Maclarin Lakim, Vincent Merckx and others, the unique biodiversity of Mount Kinabalu has evolved rapidly through a complex arrangement of long-distance colonization and local adaptation. We will jointly publish these results in a scientific journal, but Sabah Parks has also agreed to assist us to narrate this evolutionary story to the hundreds of thousands of international visitors who visit the park annually by designing exhibits to be placed throughout the park. To instill in the general public an understanding and appreciation of evolution as a process and evolutionary biology as a science can be achieved in other ways too. The citizen science project Evolution Megalab, an activity for which Naturalis managed the Dutch branch, has enlisted the help of almost 6,500 parabiologists, many of them school children, in 15 European countries to show ongoing contemporary evolution in snail shell color throughout Europe. And we published this in the journal PLOS ONE. Since then, the Evolution Megalab website and teaching materials have continued to be used in schools, sometimes with scientifically important results. For example, via the Leiden-based foundation Leven DNA and funding from Naturalis, the high-throughput sequencing facilities of the Leiden University Medical Center have allowed Ken Krijeveld and me to sequence several CPA transcriptomes, which high school students of Lisbeth Pronk's class at the Almere College in Dronten then analyzed for their profielwerkstuk. One of them even went on to become a promising student of bioinformatics at the Leiden University for Applied Sciences. Another way to enhance evolutionary literacy is through popular writing. Exactly 20 years ago, I wrote my first popular science article for Mare, the Leiden University newspaper. It was a review of the very first Naturalis exhibition, Super Insecten. And for me, it was the beginning of a long and ongoing parallel career as freelance science journalist for newspapers, magazines, websites, and more also in the form of books. Naturalis and the IBL are blessed with several other specimens of this rare species, research biologists who also engage the general public. Freek Vonk, of course, but also Lars van den Hoek-Oostende, Katja Pijnenburg, Chris Jacobs, Anne Schulp, and several other colleagues of mine are actively engaged in popularizing science. Allow me to dwell on the intersection of fundamental science and science popularization for a few more minutes. A common metaphor for the gulf between science and the general public is the ivory tower. Scientists who try to bridge that gulf are often said to descend from that ivory tower of high science to the public's level. But one could also portray such scientists as dropping rope ladders down from their ivory towers and inviting the outside world to climb up and look around. The distinction, I think, is important. Let us call the two alternatives the climbing down and inviting up approaches to science communication. Scientists who climb down often have a patronizing attitude to the general public. They carve off all the conceptually challenging bits and only feed the public easily digestible morsels. Tasty morsels, no doubt, but ultimately distracting from the core questions. Questions from which the scientists themselves get their chief motivation, but which they deem too lofty to be shared with the public. It is also these same climbing down scientists who, when asked about the relevance of their work for society, hide their true curiosity-driven motivations and instead blabber about direct but far-fetched practical applicability. I much prefer an inviting up attitude. Science is a human enterprise and as such is ultimately understandable by any interested human, regardless of their level of education. Whenever I write or speak for a general public, I make an effort not to dumb down my message, but rather to make the audience smart up. This is why in my popular science books on speciation or ecology or sexual selection, I do not shy from delving into the theory of hybrid zone analysis or food web stability or population genetics. We often fear such subjects are too complex for the general public, but the truth is that very few fields of science are resistant to popularization. As Albert Einstein said, if you can't explain it to a 6-year-old, you probably don't understand it yourself. I also have a more personal reason to profess this attitude to science communication. My father, and his father before him, hailing from humble backgrounds in The Hague and never having been near a university, were avid amateur scientists. In their spare time, they enjoyed pursuing chemistry, computer science, precision engineering, electronics, and mineralogy. They wrote on their hobbies in club newsletters and gave lectures and demonstrations for kindred spirits. It was my father who encouraged me to indulge in my eclectic childhood interests, ranging from collecting dried bird feet to turning my room into a chemistry lab, and who was so happy that I had the university education that had been out of reach for him. All too often we are under the misapprehension that the general public thinks that science is boring and useless. Hence the sometimes desperate attempts at making science fun and coming up with excuses about useful applications for basic science. I think this is a slippery slope towards becoming science apologists. Let's not feel embarrassed about our intellectual riches. In truth, society needs us in that ivory tower. To make this point for evolution and biodiversity science, hundreds of millions of people watch nature documentaries by the BBC or National Geographic Channel or the Discovery Channel. Each minute of nature documentary footage is based on years of painstaking fundamental research by a scientist somewhere, sometime. People love to learn. They love to hear true stories about the world we live in, and we scientists are the ones who do the hard research labor to provide this knowledge for the rest of mankind. Jointly, we build on our collective human knowledge, which ultimately is a source of cerebral entertainment and deep fulfillment for all. In the past years, Naturalis has already made great strides towards communicating science in the inviting up way. Literally almost in its live science hall on the 4th floor, where visitors get to interact with real scientists, watch real dissections, and can look over the shoulders of the digitization and data mining of real specimens. I am extremely happy that when the exhibitions of Naturalis are rebuilt under the guidance of Caroline Breunesse and her team, this life science hall is foreseen to be maintained and even expanded and made freely accessible to the public. I wish to end by thanking the Board of Governance of this university and the Board of Governance of the Faculty of Mathematics and Natural Sciences for the appointment, and the directorates of Naturalis and IBL for encouraging it. I am confident that with this position, interaction between Naturalis and the university will be intensified. Already, we have the focus group Character Evolution with some 60 members consisting of an even mix of IBL and Naturalis students and researchers. In several student courses, university and Naturalis lecturers closely collaborate, and multiple one-to-one collaborations exist. However, I think there is room for further expansion. The focus on model organisms within the IBL and on biodiversity within Naturalis could be capitalized upon in a more collaborative setting. The more than 100 scientists working at Naturalis hold a vast knowledge on diversity, taxonomy, and morphology, which may be tapped for comparative studies, especially given the backup provided by the fifth largest natural history collection in the world. Conversely, the IBL's expertise in experimental work and the available laboratories, such as the phytochemistry and Evo-Devo labs, may be invaluable for testing hypotheses generated by Naturalis' researchers and research groups, or vice versa. In all, I believe we stand at the threshold of a time of new blossoming of biology in Leiden. I also wish to take the opportunity to thank those on whose shoulders I stand. From my first steps along the road of science, several people stand out in having been a source of encouragement and inspiration. First and foremost, of course, my late father. I have already mentioned him. Second, my biology teacher at the Stedelijk Gymnasium in Schiedam, entomologist, environmental activist, and all-round ecologist Daan Vestergaard. As an MSc student, Dr. Schilthuizen, who supervised my work at University Pertanian, Malaysia, on tropical agricultural entomology, which lay the foundation for an ongoing tropical ecology connection with Malaysia. When I was a PhD student at the erstwhile AAW, Edi Gittenberger, Paul Breakfield, and Jacques van Alphen raised me as an evolutionary biologist. Later in Wageningen, Richard Stouthamer and Rolf Hoekstra not only were a constant source of light-hearted motivation, but also gave me a solid footing in evolutionary genetics. Furthermore, I must, from the bottom of my heart, thank my mother, my partner Rachel, and my children Fenna and Jan for always helping me feel at home in this world. Also, I wish to thank the people of my second home, the Federation of Malaysia, here represented by Her Excellency the Ambassador, for having given me for so many years the opportunity to live and work among its immensely rich tropical biodiversity and to interact with its students, scientists, and conservationists. The money that members of the audience have donated on the occasion of this inaugural lecture, and also on the occasion of Liu Torsung's PhD defense last week, will benefit the NGO Rimba in Terengganu, one inspiring group of young local researchers and conservationists, run by my colleague Ruben Clemens. Saya mengucapkan terima kasih kepada itu negara yang indah, melalui semua warga negara Malaysia yang hadir di Dewan Besar ini. And finally, I would like to address the students. You have no idea how important you are. As a constant, critical, but ultimately appreciative audience. As a soundboard for new ideas. As eager and good-humored young, hands-on researchers, always willing to test a wild idea against the real world, or to give a fresh glimmer to dusty old notions and systems. Or simply as a pleasant, promising presence, as the next generation of biodiversity researchers which, through interaction with myself and other researchers at the IBL and Naturalis, have been and are being evolved. Ik heb gezegd. Wilt u mij volgen? Kijk uit voor de trap.